It hits you in a weird way, doesn't it? The fact that you, me, even that fruit fly buzzing around your kitchen, we all trace back to the same ancient ancestor billions of years ago. Okay, let's unpack this incredible story. This deep dive is going straight to the source of it all, the origins of life, and, well, the wild ride of evolution that got us here. We've got some fascinating insights pulled from a set of research papers. Think of it as kind of a cheat sheet, some seriously deep science. And our goal is to make the truly mind-bending parts, clear and engaging. Consider this your personal expedition through billions of years, uncovering the pivotal moments and surprising turns in life's epic tale.
It's truly captivating, yeah. We're diving into questions that have, well, puzzled humanity for ages. We're starting right at the beginning, setting the cosmic and planetary stage for life. Imagine this. About 14 billion years ago, the Big Bang sets everything in motion. Then, roughly 10 billion years later, our solar system starts to form. From this massive cloud of gas and dust around, 4.6 billion years ago, early Earth is basically a blue marble dominated by a vast ocean. And it's within this primordial soup that the first sparks of biochemical systems somehow ignited.
And it wasn't quick, right? Not like flipping a switch.
Not at all. But by about 3.5 billion years ago, we think life at the cellular level was established. And interestingly, this roughly coincides with when the first land masses started appearing.
And the evidence for this is genuinely cool, isn't it?
Oh, yeah. Fossilized microbes resembling bacteria found in Australia dating back an astonishing 3.4 billion years.
Wow. But even before those, proper cells, there was probably something else, a precellular phase.
Exactly. It's likely there was. Picture self-replicating molecules, maybe early versions of RNA, or even something simpler, acting as the original blueprints of life, before cells as we know them.
So that leads to the big question. How do you actually get from just a random collection of molecules to something that can replicate and evolve?
Right. Let's look at the ingredients first. Surprisingly, the early oceans. Their salt composition was actually quite similar to today's, but the atmosphere, dramatically different. Very little oxygen, likely rich in gases like methane and ammonia, sort of soupy.
And that's where Miller-Urey comes in, that famous experiment.
Precisely. 1952, they simulated these early Earth conditions in a flask, water, methane, ammonia, hydrogen, and they used electrical sparks to mimic lightning.
And they got amino acids.
They did. Glycine, alanine, the basic building blocks of proteins. And what's even more amazing, a reanalysis back in 2007 found they'd actually made over 20 different amino acids.
Wow. Okay, so that showed the basic components could arise naturally, ebiotically.
Yes. A key insight. It showed you could get the building blocks of life from non-living stuff under plausible conditions.
Okay, so the raw materials were there. But proteins are one thing. What about RNA and DNA? Getting those building blocks, the ribonucleotides, seemed like a bigger hurdle.
It did for a while, yeah. Forming the sugar and the base and then sticking them together with a phosphate, it looked complicated. But recent research, it's shown that complete ribonucleotides can actually form more directly using precursors like cyanamide, cyanoacetylene, glycolaldehyde, glyceraldehyde, and inorganic phosphate.
So you mean you don't necessarily need to build the parts separately first, they can kind of self-assemble?
In a way, yes. They can come together more directly under the right conditions. It really shifts our understanding of how RNA's components could have emerged relatively easily.
Okay, that's a big step. So you have the ribonucleotides. How do they link up into chains, into RNA?
Well, it's thought that base stacking interactions could have played a role. You know how the flat bases of RNA kind of stack like coins? There's a weak attraction there. Individually, it's weak. But over long chains, or maybe concentrated on surfaces like clay, or even ice, or maybe through cycles of wetting and drying, like in little ponds evaporating and refilling, or maybe even in atmospheric clouds. These interactions could have helped link them together. Polymerization.
So plausible pathways exist chemically and geologically.
Plausible pathways, yes, for both the building blocks and maybe even linking them up.
But, like the research says, that massive leap from just a messy soup of molecules to something actually organized and lifelike.
That's still pretty theoretical, right? We haven't quite nailed that in the lab.
Not fully, no. That specific transition remains speculative experimentally. It's a huge jump in complexity.
But then you think about the scale. The early oceans were vast, maybe 10 billion biomolecules per liter.
Right.
And the time involved, maybe a billion years for this stuff to potentially happen.
Exactly. Over those kinds of scales, even incredibly improbable chemical events become, well, much more likely. Almost inevitable, perhaps. Which brings us to that classic chicken and egg problem, doesn't it? The polynucleotide polypeptide problem.
Ah, yes. The dilemma. Proteins are great catalysts. They do all the work in cells, but they can't copy themselves. Polynucleotides, like RNA and DNA, they carry the information. They can be copied, but we initially thought they couldn't really do much catalytically without proteins.
So you need proteins to make polynucleotides and polynucleotides to make proteins. How could life ever start? It sounds like a paradox.
It did seem like one until the 1980s.
Ribozymes.
Exactly. The discovery of ribozymes, RNA molecules that act as enzymes. That was the game changer.
And they can do some pretty amazing things, right? Cut themselves, cut other RNAs.
Even form peptide bonds. That's the core reaction for making proteins, and it's catalyzed by RNA within the ribosome in all life today.
So RNA could be both the blueprint and the builder, potentially.
Precisely. It blew the doors open. Suddenly, you didn't need proteins and nucleic acids to arise simultaneously in all their complexity. RNA could have come first. And scientists have even engineered synthetic ribozymes in the lab that can do other key things, like make more ribonucleotides, copy RNA templates, even link amino acids together.
And then there are riboswitches, too, RNA regulating itself.
Yes. RNA molecules that change shape and function when they bind to small molecules, acting like little genetic switches. All this evidence points towards the immense versatility of RNA and strongly supports the
RNA world hypothesis.
The RNA world hypothesis, the idea that early life, or at least the precursor stages, was dominated by RNA.
So how does that work? RNA copying itself probably wasn't very good at it initially.
Likely not. Probably slow, very error-prone. Like, making a photocopy of a photocopy repeatedly, it gets messy.
But maybe the messiness was actually good.
Paradoxically, yes. Those errors would generate variation, a diversity of RNA sequences. By pure chance, some of those variants might have stumbled upon slightly better catalytic activities. Maybe one could copy itself a tiny bit faster or a little more accurately.
Ah, so natural selection kicks in, even at the molecular level.
Exactly. Those slightly better replicators would start to outcompete the others. And as replication became more accurate over time, it allowed for longer RNA molecules to evolve.
Longer RNA means more potential for complex folding and more sophisticated functions.
Precisely. Like the large ribosomal RNAs we see today, which are complex molecular machines, you can imagine a sort of protogenome emerging, maybe a collection of self-replicating RNAs directing some simple biochemical reaction.
Would be even basic energy reactions, like using ATP.
Possibly. Hydrolyzing ATP or GTP for energy is fundamental, and maybe these systems got packaged up.
Inside membranes, little bubbles of fat.
Lipid membranes, yeah, forming the first protocells. Compartmentalization would be a huge advantage, keeping all the useful molecules together.
It paints a really compelling picture. But while we don't live in an RNA world now, we live in a DNA protein world. How did that transition happen?
That's the next crucial chapter. Protein enzymes started to emerge. And proteins, with their 20 different amino acid building blocks, turned out to be much more versatile catalysts than RNA generally is.
More chemical variety, more complex shapes.
Exactly. They could perform a wider range of reactions and often much more efficiently, so they gradually took over most catalytic roles from ribozymes.
Why did they evolve, maybe being stuck inside those lipid vesicles?
That's one idea. RNA is generally hydrophilic, water-loving. Membranes are hydrophobic fatty. Maybe proteins evolved partly as a kind of interface, a hydrophobic coat needed for the RNA machinery to function within or interact with the membrane.
Interesting. So the job of the RNA shifted from doing the chemistry to coding for the proteins that do the chemistry.
Right. The protogenomes became less about direct catalysis and more about holding the instructions for building catalytic proteins.
And the research makes a neat point there. It wasn't necessarily the ribozymes themselves becoming coding sequences, but more likely they synthesized the molecules that became the coding sequences.
Yes, that seems more plausible. A gradual handover of function.
And then the switch to DNA. Why DNA?
Stability. DNA is just chemically much more robust than RNA. That little difference in the sugar deoxyribose versus ribose makes DNA less prone to breaking down, especially in the long strands needed for larger genomes.
So it's better for long-term information storage.
Much better. And the building blocks, the deoxyribonucleotides, can be made from ribonucleotides relatively easily. You might have even had early enzymes, sort of like reverse transcriptase, making DNA copies from RNA templates.
And swapping uracil for thymine. That helps too.
It does. It makes DNA repair more efficient. Cells can easily spot and fix a common type of damage, cytosine, turning into uracil because uracil doesn't normally belong in DNA. Thymine gives it that unique identity.
Plus being double-stranded. That offers a backup copy for repair too.
Absolutely. Double-stranded DNA is far more stable and allows for sophisticated repair mechanisms. The move to DNA was really key for enabling larger, more complex genomes to evolve.
But early genomes probably weren't the neat chromosomes we have now.
Likely not. Probably more like a collection of many separate, smaller DNA molecules. Maybe single genes or small groups of genes.
Like scattered notes.
Yeah. Organizing them into larger chromosomes came later, likely because it made cell division, distributing all that genetic information accurately to daughter cells, much more efficient. From notes to chapters in a book, as you said.
Okay, so we have this picture of RNA giving way to DNA and proteins. But did this whole process happen just once? Or could life have started independently multiple times?
That's a fascinating question. The initial steps, maybe forming amino acids or simple polymers, could potentially have happened in many places on early Earth. But the evidence strongly suggests that all life we know today descends from a single common ancestor, LUCA, the last universal common ancestor.
And that crucial step was probably when one system nailed the DNA-RNA protein combo.
That seems most likely. Once one lineage developed protein synthesis directed by a nucleic acid code and adopted the stability of DNA, it would have had a huge advantage.
Better catalysts, more stable information storage, more accurate replication.
Right. These cells could likely reproduce faster, more reliably, maybe utilize resources more efficiently. They probably would have outcompeted any lingering RNA-based life forms, maybe even, consume them. Evolutionary selection favoring the more robust system.
It's pretty decisive. But it does make you wonder, could life have gone down a different chemical path entirely, using different building blocks?
A truly intriguing possibility, and the research delves into the world of alternative nucleic acids. For instance, they mention an alternative base pair involving purine 2-poly-6-decarboxylate and 3-pyridine held together by a copper ion.
A copper ion, not hydrogen bonds.
Exactly. And amazingly, some DNA polymerases, the enzymes that copy DNA, can actually incorporate these weird bases in the lab.
That's wild. It shows the AT-GC pairing isn't the only possibility.
Not chemically, no. And it's not just the bases. The backbone could be different too. The research mentions PNA, pyrinosyl nucleic acid. It uses a 6-carbon sugar instead of RNA's 5-carbon one, and it actually forms more stable base pairs than RNA does.
More stable. Wow. And others.
There's TNA, threose nucleic acid with a 4-carbon sugar, and GNA, glycerol-derived nucleic acid, which has a simpler linear backbone, no sugar ring at all.
So the fundamental structure could vary a lot.
It could. And what's really interesting is that these synthetic alternatives can often form stable hybrid structures with natural RNA or DNA.
Which suggests maybe they played a role in early life. Or maybe life elsewhere uses something like this.
It certainly broadens our perspective on what life could be chemically. It's not necessarily limited to the DNA system we know. Fascinating area for astrobiology, too.
It really does open up a universe of possibilities.
Okay, let's fast forward a bit. Billions of years pass, life diversifies incredibly. The research gives a kind of highlight reel of major morphological steps. Right, a timeline of milestones. You have bacteria firmly established by 3.4 billion years ago. Then the first hints of eukaryotes, cells with complex internal structures like a nucleus, show up as chemical signatures, sterols, around 2.7 billion years ago.
But the first actual visible eukaryofossil, Bangiomorpha, that's much later.
Much later, yeah. About 1.2 billion years ago. Then you get the first clear signs of multicellular animals around 640 million years ago. Big jumps in complexity.
And then the famous Cambrian explosion.
Around 530 million years ago, yeah. This burst of diversification where most major animal body plans appear quite rapidly in the fossil record, though it was followed by a major extinction event not long after, around 490 million years ago.
Evolution isn't always a smooth ride.
Definitely not. Later on, you see life conquering land insects, animals, plants establishing terrestrial ecosystems by about 350 million years ago. Then the dinosaur extinction 65 million years ago, opening the door for mammals. And finally, our own lineage, the first hominins, appearing around 4.5 million years ago.
It's just staggering timescales. And running alongside this morphological evolution, there's a general trend in genome evolution, isn't there? Increasing complexity.
Generally, yes. There's a correlation. As organisms became more complex morphologically and functionally, their genomes tended to get larger and accumulate more genes. Think about it going from fewer than 1,000 genes in a typical bacterium to around 20,000 genes in vertebrates like us. That's a huge increase in genetic information.
So where do all these new genes come from? How does a genome get bigger?
There are really two main ways genomes acquire new genes. First, duplication of existing genes making extra copies. Second, acquiring genes from other species, lateral gene transfer, or LGT, both provide the raw material for evolution to work with.
Okay, let's tackle gene duplication first. So a gene gets copied. Now you have two identical versions. What happens next?
Well, initially they're identical. The research points out there's usually strong selective pressure to keep at least one copy functional, doing its original job. The organism still needs that function.
But the second copy, it's kind of redundant, isn't it? So it's free to change.
Exactly. It's under relaxed selection. And it faces a few possible fates. It might just stick around doing the same job, which can be good if having more of that gene's product is beneficial, a dosage effect. More often, though, because it's not essential, it starts accumulating random mutations. These mutations often disable the gene.
Turning it into a pseudogene, a non-functional relic.
Precisely, a non-processed pseudogene. That's actually the most common outcome for a duplicated gene. It becomes a sort of genetic fossil.
But sometimes those mutations might actually create something new and useful.
That's the exciting part. Occasionally, random mutations can tweak the duplicated gene's function in a way that turns out to be beneficial for the organism. If this new function provides a survival or reproductive edge, natural selection will favor keeping that altered copy. And boom, the genome has gained a new functional gene, increasing complexity. It's evolution tinkering.
And when you have families of related genes, like these duplicates, how do they stay similar when needed or coordinate changes? The research mentions concerted evolution.
Right. Sometimes it's advantageous to keep multiple copies of a gene very similar, or to spread a beneficial mutation through all copies. Concerted evolution is the process that homogenizes gene families over time. The likely mechanism behind it is gene conversion. It's a process where one DNA sequence replaces another homologous sequence. So one gene copy can essentially overwrite part of another copy, keeping them similar or spreading changes. This is especially common for genes arranged close together in tandem arrays.
Okay, let's make this concrete. Examples. The globin genes are a classic one, right?
Right, perfect example. All the globin genes coding for proteins like hemoglobin in blood, myoglobin in muscle, neuroglobins in the brain, they all trace back to a single ancestral globin gene that started duplicating way back, maybe 800 million years ago.
So one gene duplicated, then those copies duplicated, and they diverged.
Exactly. They diverged, accumulating mutations that specialize their functions. Now we have globins optimized for oxygen transport in different tissues, or even at different developmental stages, like fetal hemoglobin versus adult hemoglobin. That's a clear case of duplication followed by neofunctionalization, gaining new functions.
And we can even estimate when these duplications happened using the molecular clock concept, looking at the accumulated differences.
Yes, by comparing the sequences in different species and knowing roughly how fast sequences change, we can build evolutionary trees and estimate the timing of these duplication events.
What about the beta globin cluster in mammals?
Another great example, showing more recent and dynamic evolution, if you compare the cluster of beta-globin genes in, say, humans, rabbits, and goats, you see a complex pattern of recent duplications, some genes becoming pseudogenes, gene conversion events, homogenizing parts of genes, even fusions between genes. It's a snapshot of evolution in action over shorter timescales.
And then the Hox genes. These are crucial for body plans, right?
Absolutely critical. Homeotic selector genes. They tell different segments of the developing embryo what to become: head, thorax, abdomen, where limbs should go, master regulators.
Fruit flies have just one cluster of these?
One HOMC cluster, yes. Probably arose maybe 1 billion years ago. But most vertebrates, including us, have 4 Hox clusters, usually on different chromosomes.
4. So that suggests
2 rounds of whole genome duplication in the early vertebrate lineage. Those duplications copied the entire ancestral Hox cluster twice.
and having more Hox genes allowed for more complex body plans to evolve.
That's the strong inference. More genes provide more regulatory control, potentially enabling the evolution of more intricate anatomical structures. And it doesn't stop there. If you look at teleost fish, they generally have 7 or 8 Hox clusters. And salmonids, like salmon and trout, have even more, maybe 13.
Whoa, more duplications.
More rounds of duplication, yes, correlating with diversification in those fish lineages.
Yeah.
It's a powerful link between gene duplication, genome evolution, and morphological complexity.
Okay, so how do these duplications actually happen, mechanistically?
Several ways. Unequal crossing over is a key one. During meiosis, when chromosomes pair up, sometimes they misalign slightly, especially if there are repetitive sequences. If crossing over, the exchange of DNA happens in that misaligned region, one chromosome can end up with a duplicated segment and the other gets a deletion.
Like swapping unequal bits of information.
Pretty much. A similar thing, unequal sister chromatid exchange, can happen between the two identical copies of a chromosome after it replicates but before the cell divides. There's also DNA amplification through replication errors, which can generate multiple copies. The common theme is usually recombination happening between short, repeated DNA sequences, leading to the duplication of the DNA in between them, often initially creating tandem duplicates copies right next to each other.
But genes don't always stay next door after duplication, do they? You mentioned aldolase genes are scattered. How do they get dispersed?
Right. Sometimes large-scale chromosome rearrangements, inversions, translocations can move duplicated genes around. But another really important mechanism for dispersal is retrotransposition. This involves an RNA intermediate.
Ah, the copy and paste mechanism using RNA.
Exactly. A gene gets transcribed into messenger RNA, mRNA. Then, an enzyme, usually reverse transcriptase, encoded by a mobile element like a line retrotransposon, makes a DNA copy, cDNA, from that mRNA. This cDNA copy can then integrate back into the genome pretty much anywhere else.
And what happens to that new copy?
Often it lands somewhere without the necessary control signals, the promoter, to turn it on. And because it was made from mRNA, it lacks introns. These usually become inactive process pseudogenes.
But if it lands near an existing promoter by chance...
But then it can become an active gene again. It's called a retrogene. Still no introns, but functional.
And sometimes the original gene gets lost, leaving only the retrogene.
Yes, those are called orphan retrogens. Humans have maybe 600 or 700 identifiable retrogenes in total, and perhaps 25 or so are orphans. Retrotransposition is a major way gene families get dispersed across chromosomes.
Okay, going back to duplication for a moment. Sometimes the point isn't to evolve a new function, but just to have more copies of the same gene, right? Like for dosage.
Absolutely. The research uses RNA genes, the genes for ribosomal RNA, as the prime example. Ribosomes are the protein factories, and cells, especially rapidly growing ones, need tons of them. Making all that RNA requires having many, many copies of the RNA genes working simultaneously.
So it's about production capacity. How many copies are we talking?
Hundreds in humans, maybe 350 or so. Some plants, like peas, have thousands, over 4,000 copies.
Wow. And for these, you don't want them diverging. You want them all making identical functional RNA.
Exactly. This is where concerted evolution and gene conversion are vital. They act to keep all those hundreds or thousands of copies essentially identical over evolutionary time, weeding out mutations and homogenizing the sequences. It's selection for maintaining identity, not divergence.
So we've covered whole gene duplication, but evolution can also tinker with just parts of genes,
Yeah.
duplicating domains.
Yes, domain shuffling or exon shuffling. This is another really elegant way to create novelty. Many proteins are modular, built from distinct functional domains encoded by exons. Evolution can duplicate and rearrange these exons, essentially mixing and matching functional units to create new proteins with combined or altered functions.
Like protein Legos?
Kind of, yeah. The research gives the example of TPA, tissue plasminogen activator. Its gene is a mosaic containing exons that encode domains clearly related to domains found in other proteins. A finger domain from fibronectin, a Kringle domain from plasminogen, a growth factor domain from EGF. Combining these created a protein with a specific function in dissolving blood clots.
And collagen, with its repeating units.
Another good example of domain repetition contributing to protein structure and function, in collagen's case, strength and flexibility. Domain shuffling is a powerful route to protein innovation.
Okay, so we've got duplication within a species. What about stealing genes from other species? Lateral gene transfer. How common is that?
It's incredibly common in prokaryotes, bacteria, and archaea. They swap genes readily. Conjugation is one way direct transfer, often via plasmids or transposons, usually between related species. Transformation is another, picking up naked DNA from the environment. This can happen even between very different species. It really blurs the species boundaries in the microbial world.
What about plants and animals?
Plants can do it sometimes, particularly through hybridization followed by genome doubling allopolyploidy. That brings together genes from different species. But in animals, LGT seems to be extremely rare. There are a few suspected cases, maybe mediated by viruses or transposons carrying genes across species barriers, but it's definitely not a major driver of gene acquisition in animals the way it is in bacteria.
Okay, there's one more big mechanism for increasing gene number we need to cover. Doubling the entire genome. Polyploidy. That sounds like a massive event.
It's huge. The most dramatic way to instantly increase gene number across the board. There are two main ways it happens. Autopolyploidy is basically an error within a species meiosis messes up, producing gametes, sperm or egg, that are diploid 2N instead of haploid N. If a 2N gamete fuses with a normal 1N gamete, you get a triploid 3N offspring. Allopolyploidy involves hybridization between two different species. The hybrid offspring might be sterile, but if its entire chromosome set then doubles, it can restore fertility and create a new polyploid species carrying genomes from both parents.
Triploids are often sterile, though. Problems pairing chromosomes.
Often, yes. That odd number of chromosome sets makes meiosis difficult, but if the genome doubling occurs, creating a tetraploid 4N, for instance, meiosis can proceed more normally.
And the big evolutionary consequence is massive gene duplication.
Exactly. Instantly, every single gene has at least one extra copy. This provides enormous redundancy. Just like with single gene duplication, these extra copies are freed from constraint. They can accumulate mutations, diverge, take on new functions, or be lost over long evolutionary timescales. It provides a huge burst of raw genetic material for evolution to experiment with, potentially fueling major evolutionary transitions and diversification.
And we can see evidence of ancient polyploidy events in genomes today.
We can. Molecular clocks often show large sets of duplicated genes, all appearing to originate around the same time, pointing to a whole genome event. We also see synteny large blocks of genes that are found in multiple copies, often in the same relative order, on different chromosomes. That's a telltale sign of ancient genome duplication.
Where have these events happened?
Lots of places. The research mentions yeast had one about 100 million years ago. The model plant Arabidopsis has undergone multiple rounds. We already talked about the two rounds, called 1R and 2R, early invertebrate evolution that duplicated the Hox clusters. And then there was another round, 3R, in the ancestor of teleost fish, and further rounds in specific fish groups like salmonids.
So it's happened repeatedly in different lineages. Phylogenetic trees must show this.
They do. By mapping gene duplications onto evolutionary trees, researchers can pinpoint inferred whole genome duplication events that likely spurred diversification in vertebrates, various plant groups like legumes and grasses and other lineages. It's clearly been a major engine of evolution.
Okay, so whole genome duplication is the big one. But the human genome also has lots of smaller, more recent duplications, segmental duplications.
Yes, our genome is riddled with relatively recent segmental duplications, or SDs, sometimes called low-copy repeats, LCRs. These are chunks of DNA, anywhere from 1 kilobase up to maybe 400 kilobases long, that have been copied. They have very high sequence identity, usually over 90%, meaning they duplicated recently. They might be present in just 2 copies, or maybe up to 50 or so times.
How are these different from things like lines and signs?
The copy number mainly. Lines and signs are present in hundreds of thousands or millions of copies. SDs are much lower copy number, though the duplicated segments themselves can certainly contain lines and signs within them. Interestingly, comparing human and mouse genomes, humans seem to have a higher proportion of very recent, highly similar SDs, both within chromosomes and copied between different chromosomes. It suggests our genome has been particularly dynamic in this respect recently.
It's just amazing how fluid genomes are. Always changing. Now, we focused a lot on the coding DNA, the genes, but that's only like 1.5% of the human genome. What about the rest, the non-coding stuff?
Right, the vast majority. Generally, non-coding DNA evolves differently because it's under less direct selective pressure to maintain a specific protein sequence. This means mutations tend to accumulate faster in most non-coding regions compared to coding regions. They diverge more quickly between species.
But it's not all just junk, right? Some non-coding DNA must do something.
Absolutely not all junk. We now know large parts of it are transcribed into RNA, and many regions contain crucial regulatory elements that control when and where genes are turned on or off. Introns, the non-coding sequences within genes, have to be spliced out correctly. Transposons, even though often seen as parasitic, can sometimes acquire functions or influence nearby genes. The evolution of non-coding DNA is complex, with different parts evolving under different constraints. It's a very active area of research.
And you mentioned transposons, the jumping genes. They don't just sit there. They can actually cause more changes.
Definitely. They're major agents of genome rearrangement. Because you often have many similar copies of transposons scattered around, they provide targets for recombination. Illegitimate recombination between transposons in different locations can lead to deletions, duplications, or inversions of the DNA segments between them.
So they can scramble the genome. Usually bad, but maybe sometimes good.
Often detrimental, yes. But occasionally such a rearrangement might be beneficial. The research mentions an example where line 1 recombination might have been involved in a beta-globin gene duplication about 35 million years ago. Plus, the act of transposition itself has effects. A transposon inserting into a gene can disrupt it. Inserting near a gene can alter its expression. Some transposons, like line 1s, can accidentally carry adjacent genomic DNA along with them when they move. That's called three-put transduction. They can even shuffle exons around if copies exist within introns of different genes. They really stir things up.
Okay, one last major topic the research touches on. Introns, those non-coding bits inside genes. Where did they come from? It's been debated for ages, hasn't it?
Since 1977, yeah. A long-standing puzzle. There are basically two main camps.
Introns early versus introns late.
Exactly. The introns early hypothesis suggests introns were present in the very earliest genes, maybe even back in the RNA world or just after, and that organisms like bacteria, which mostly lack introns, lost them later. This fits nicely with the exon theory of genes. The idea here is that early genes were assembled from smaller minigenes, each coding for a small protein domain or functional unit. These minigenes became the exons.
And the DNA bits linking them together became the introns.
Right. Then the splicing machinery evolved to cut out the intron transcripts and stitch the exon transcripts together, allowing the creation of larger modular proteins from these ancient building blocks.
So introns are ancient relics of how genes were first put together.
That's the intron's early exon theory view. It suggests the exon-intron structure is fundamental and ancient, at least in the lineage leading to eukaryotes.
And the intron's late idea.
That introns invaded eukaryotic genomes later, perhaps spread by mobile elements like transposons, inserting themselves into pre-existing intronless genes. The evidence is complex and the debate continues, but the structure here leans towards introns early.
Fascinating. Okay, let's try to wrap this incredible journey up. We've covered a huge amount of ground.
We really have, from the Big Bang and the first molecules through the possibility of an RNA world.
The crucial shift to the more stable DNA and versatile proteins.
The immense power of gene duplication and even whole genome duplication as engines for creating new genetic material and complexity.
The sideways movement of genes via lateral transfer, especially in microbes.
And the dynamic nature of the entire genome, including non-coding regions and those disruptive but sometimes creative transposons. It really underscores how evolution works by tinkering, copying, rearranging, and selecting.
And yet, despite all we've learned, there are still huge unanswered questions, aren't there?
Oh, absolutely. The precise step-by-step transition from non-living chemistry to the first replicating cells that abiogenesis gap is still a major puzzle. And fully understanding the functions, if any, of all the non-coding DNA, the so-called dark matter of the genome, is an ongoing challenge. There's still so much to discover.
But stepping back, understanding this Jeeve history, it gives you a different perspective, doesn't it, on how connected everything is?
It really does. That shared ancestry stretching back billions of years, it connects us to every living thing on the planet. We're all variations on a theme that started incredibly long ago. Thinking about the sheer timescale, the contingencies, the near accidents, the selective pressures that shaped life, it's profoundly humbling.
Absolutely. So maybe a final thought for everyone listening. Given all the randomness, the duplications, the rearrangements, this constant evolutionary churning, what truly unexpected biological forms or functions might still emerge in the future here on Earth? Or perhaps what completely different forms might life have taken elsewhere in the universe, based on entirely different chemical principles? Lots to think about. Thanks for diving deep with us today.